I'm Peter Donnelly. I'm now Chief Executive Officer and one of the founders of Genomics plc. Early on in my career and my work, I was really driven by the mathematical challenges, which were fascinating and really interesting in the work I was doing, which was about modeling the way populations evolved. And I came to realize actually that the scientific questions were even more interesting than the mathematical ones. Towards the end of the 1990s, which is when this transition was happening for me, that was the time when experimental technologies were developing, which could generate quite large amounts of data on individuals from populations, humans and other species. And there was a real need to develop sophisticated statistical tools to really make sense of that extra level of detail, extra level of information. I happen to be really well placed for that because of my mathematical and statistical background. And so I was lucky enough to be involved in tackling some of those early problems. Our work in Genomics plc is about trying to harness the power of genetics and genomics in improving healthcare. So at the moment, genetics typically plays into healthcare in one area, what's called genomic medicine, which is about people with rare diseases, typically caused by a single or in some cases two changes in their DNA, which make them really ill. But actually we see a world in which genetics goes from just that area to being routine within healthcare. There are massive differences in people's disease risk based on their genetics because of many, many, many different DNA letters that they carry. So we've developed the technology to measure those individual positions. Different ones are important for different diseases. There are up to 1 million or more positions which matter for each of the diseases. And we combine that information to get something called a polygenic risk score. We can do a single genetic assay, a genotyping assay, so measure someone once, get the genetic data from that, and then calculate polygenic risk scores for many, many different diseases. So that allows us to identify people who are at high risk for different diseases in ways that currently those individuals are invisible to healthcare systems. This idea of predictive genomics, of using genetics to understand people's risk for common diseases, of changing the way we do prevention in healthcare to harness that, it's totally new. So it's not as if there's an existing need and we've got a better way of filling it. It's about re-engineering healthcare systems to do things in a more sophisticated way. And that's a major focus for us, but it's a chasm from the science to making a difference in healthcare. It's incredibly rewarding. If we're successful and if others doing the kinds of things we're aiming to do are successful, there'll be millions of people alive in 20 or 30 years' time who wouldn't be alive otherwise. That's how important it is, but it's challenging. At a technical level, one of the challenges in polygenic risk scores is, as we've shown and many others have shown, the polygenic risk scores typically perform better. In other words, they're more powerful to predict for individuals of some ancestry groups, often European ancestry groups, than others. A lot of the problem is the imbalance in available data, that there's more data available on people from some ancestries, typically European ancestries, because that's where a lot of the early genetic studies were done. There is a move towards greater diversity, both in terms of the data sources, actually, and in terms of the scientists in the field. But there's still more to be done on both fronts. In the early 2000s, when we and others were thinking about genome-wide association studies, microarray technologies were the only available technology that would give us data on the number of individuals, the substantial number of individuals we needed in a way that was affordable. So that they were a very natural choice in the era of genome-wide association studies. My experiences with Thermo Fisher Scientific on array design have been fantastic. I have found them unbelievable partners to work with. The people on the bioinformatics side are incredibly talented, very responsive and very nice. And the colleagues on the technical side, the ones who can tell you whether you might be able to design for this SNP or that SNP, they've also been brilliant. It's fair to say that the arrays that have come out of that design process, where we've contributed a lot because we've been bringing in the information and the kinds of SNPs that we want involved, but the colleagues at Thermo Fisher have been absolutely central to it. Those arrays have been really successful. We know that genetics is an important part of human variation. It's an important part of the way human biology works. We've scratched the surface on understanding how that plays into certain aspects of healthcare. But that's the next set of challenges, to move genetics from rare diseases where it's already in healthcare through to predictive genomics and prevention, where I think it has massive potential, to the stage where it's just ubiquitous in healthcare systems and we can start re-engineering the healthcare systems to take advantage of that. I have loved being a scientist and I feel incredibly lucky. First of all, it's amazing to be able to pursue the questions that you find really interesting. Others have to find them interesting enough to fund them. Secondly, it's a fantastic privilege to work with incredibly talented people. You know, I've been lucky enough throughout my career to work with amazing people in my own research group, to have amazing colleagues within my institution, but the whole international scientific community is a really great and rewarding place to work. I think for me, the third thing that I've loved is that I love learning new things. So I've loved learning, and I've still got a lot to learn, which will keep me going for a while.